Welcome to our Friday seminar series. This may be one of the first or second graduate students that have appeared this semester. This is Maya Enriquez, who is an IBS master's student. And as part of her master's thesis, she is required to give a public seminar. So thank you for coming and giving your support. Maya came for us from Vassar College or University? College in Poughkeepsie, New York. Thank you. And she's done an outstanding job here, both as a graduate student and as a teaching assistant. And she's one of the COVID kids. So she started under normal circumstances, and then COVID hit. And the amazing thing about Maya is she came to work on toadfish in Woods Hole, Massachusetts with me, and then ended up working on blind cavefish in Minnesota. We pivoted from Woods Hole to cavefish in about 2 weeks. And it was an amazing turnaround. I really appreciate her flexibility. The nice thing is Amanda Drews actually had invited a speaker from the Twin Cities, Susan McGaw, to come up here and give a talk. And we met after that, and we noticed we both had interest in sensory physiology of fishes. And it worked out fine, because when COVID hit, I called Suzanne and asked, hey, can we switch our project? And she said, sure. So she sent us all the fish. And in fact, she's so impressed with Maya's work is that Maya should be entering or getting her PhD in the McGaw lab this coming fall. But life is not perfect, because as punishment for some things she's done before in her life, which I don't understand, Suzanne is sending her to Lehigh University for spring semester. And if you've never been to Lehigh, it's cold, black snow, freezing rain. It's probably the most hideous campus in all of Eastern Pennsylvania. But luckily, Maya's, has such a great personality. She said, "Sure, I'll go." Okay, so that's what she has to look forward to, and that was not my decision, so she can't blame me. So, without any further ado, here's Maya's thesis.
Thank you, Dr. Menzinger, for the lovely introduction. My name is Maya, and today I am very excited to share with you all the focus of my master's thesis, investigating rapid divergence of sensory systems between satellite populations of the Mexican tetra, A.mexicanus. Now, before we get started, I'd like to start my science with a story, because I feel like that really makes it come alive. So please suspend your disbelief and come with me on a story very quickly. It is difficult to imagine how imposing it is to enter those most magnificent natural grottos. One enters first through a transom-like opening just large enough for one man. After walking for about 1 kilometer through a narrow gallery obstructed in places by enormous fallen rocks, one arrives at a chamber large enough to contain a cathedral, completely covered with stalactites and stalagmites, which present a grand aspect when illuminated. At last, we came to the zone of creeks and pools. The first pool with which we encountered is so very deep that we cannot estimate its depth. After edging our way around this pool by holding onto the fissures in the rock, we continued along a creek, walking knee-deep in the water in a head-high passage, finally arriving at another large pool, also very deep, in which it was possible to see with the light the little fishes for which we had come. Now, personally, I think this is a fantastic story pulled out of an epic, but it is actually true life. Let me turn on the lights. So what you just heard is actually an excerpt of a study, actually the first publicized study where we found A.mexicanus, published by Hubbs and Ide in 1936. It is the first scientific documentation of this species, but it had a different name. Initially, A.mexicanus was known as Anophthyctus jordani, Anophthyctus meaning bony fish with no eyes, and Jordani named after the eccentric fish collector that asked for this specimen to be collected and documented. Unfortunately, today we will not be spelunking into caves or discovering exciting new species, but come with me on a journey as we learn more about the natural history and sensory physiology of A.mexicanus. So in order to keep ourselves oriented to where we are going and what we're talking about, I have a bit of a roadmap for all of us to follow. So for my seminar today, we will start with an extensive background giving you context to why we did this study. We will then narrow down and dive into the research question, really the meat of this work. We will then talk about 3 different methodologies that were used in this study. The first being auditory evoked potentials, the next one being particle acceleration, and the last methodology known as electroretinography. And at the end, we will wrap it up nicely by doing a summary of our results and future directions. So why don't we get started? Now, I think we would be remiss to talk about cave organisms and cave fish if we did not talk a bit about cave ecology. I think as all of you scientists know, organisms are a reflection of their ecologies and their environments. So let's talk a bit about caves. I'm going to be focusing on the benefits as well as the challenges of living in a cave environment. It might be a bit obvious, but caves are extreme environments, and there are quite a few challenges for organisms that live there. For one, you have perpetual darkness. Because of this perpetual darkness, there is no sun up and sun down. There is the absence of environmental cues, so circadian rhythms typically get dissociated. You also have in cave environments typically lower nutrient levels than you would above ground, and in some cases also elevated levels of CO2. All of these make it difficult to survive in a cave. However, for organisms that are able to exploit these resources and persevere, there are many benefits to living in a cave. For one, it is typically a refuge from predation. You will not get predated upon here. There are just not many organisms that live there. There's also reduced competition for resources, so you will likely not starve or have to compete for food. And also, it is a refuge from weather extremes. So while it is a static environment and it is a challenging environment, it is consistent, and that helps for organisms to adapt. Now, let's talk a little bit about cave fish. It might surprise you that there's more than one species of cave fish. In fact, shown by this phylogenetic tree, there are 10 distinct teleos families that contain cave fish, and they are all over the globe in every continent except Antarctica and are quite diverse. The top fish is a waterfall crawling species that was found in Thailand in 2016. And the bottom one is the largest cave species known currently at a whopping 3 pounds, 10 times larger than any other species known, discovered in India in 2017. There is a lot we have yet to learn about cave fish. Now, one thing you might notice when you look at this map, a lot of the places where cave fish are found overlap with these areas known as karst regions. What is a karst region? A karst region is a name for a particular geological formation, which leads me to my next slide. If we're going to be talking about caves, this is also a story of geology. Specifically, it is a story of something known as speleogenesis, which is the generation and the process which creates cave systems. Now, cave systems vary, but the ones we're going to talk about today, karst cave systems, develop when you have these limestone deposits and you have acidic rainwater. Acidic rainwater will enter these crevices, creating cracks and fissures in the rock, and sometimes water will fall through, creating these water systems in these caves. Sometimes it'll be through a sinkhole that the water comes in and a river through which it comes out, but they're quite diverse. And they can be also quite deep. Here we have an example of a sótano, which is also known as a sinkhole in Mexico. It's known as Sótano de las Golondrinas, or Cave of the Swallows, which is one of the most famous ones in Mexico. And you can see from this aerial photograph, it's quite large, but if we look at a cross section, even grander, more than 300 meters deep in some places. Just trying to hammer the point across that these cave systems can be very deep, very extensive, and very complex environments. Now that we've talked about environment, let's talk about some features that we see in cave organisms. I'm going to be talking about specifically troglomorphy. Troglomorphy describes a convergence of traits that we see in cave organisms because of the consistent environment you find across cave systems. So if we're looking at this salamander here and this ulm on the other side, you can see one difference, it's pretty obvious. It's a lack of pigmentation. Rather than having pigment and colors, it is pretty translucent and pink. And in an environment where you don't have any light, you don't really need pigment. Another thing that you'll see is this cave organism has no eyes. Typically in cave organisms, eyes are the first thing to go as they are metabolically costly. And if you're constantly in the dark, there is not much for you to see. There are other traits that can't quite be seen, but can be, when you look at their behavior, are better developed and looked at. So we have things like usually decreased aggression. We'll have a decrease of schooling behaviors and things like an increase in adipose fat tissue or an increase or change in dietary habits. So there's a lot of different things that encompass a cave organism as they adapt to this extreme environment. Now we can talk about the star of the show, which is my organism, A.mexicanus. Now, when I first pull up this slide, you might think that I'm showing two different organisms, but actually this is one species. So if we look at this fish here, it has a lot of traits that we typically associate with regular fish. They have eyes, pigmentation, body shape, all looks familiar to us. This fish, on the other hand, displays a lot of the troglomorphic features that I talked about on the other slide. They have no eyes and they have no pigmentation. So maybe you would say this appears to be two species of fish, but it is one species and two ecotypes. This means that although it is one species and can interbreed, they have two very different phenotypes as a result of their different environments. Let's dive into some scientific history of A.mexicanus. So, as I described with our excerpt and story, we have the discovery of A.mexicanus in 1936 by Hubbs and Innes. In 1947, we discovered a couple more populations, and I would say arguably the most exciting discovery of this species occurred in 1957, when Sadoglu discovered that they can interbreed and hybridize freely between cave and surface forms. This really led to an increase in studies on this species, as it is quite unique and a very good case study in evolution. In 1967, they changed the nomenclature to show that it was not Anoptictus, its own species, but actually Astyanax and just different morphotypes. In the 1970s, we discovered an additional 27 populations. And then in the 1980s and 90s, we did a little bit more work into understanding about their natural history. We looked at things like their ability to endure decreased oxygen consumptions, alteration to circadian clocks, and also some differences in behavior and phenotype. Now in the 2000s, the most recent work that we've been doing with Astyanax has to do with their genetics. Actually, most recently in 2014, the first draft of the physical draft of the genome was conducted and shown and published, which is very exciting. Yes, I agree. Since then, a lot more research has been done with A.mexicanus in medical settings, but that's going a bit beyond the scope of the study. But you should know that it is very much an up and coming research organism in the field of medicine. All right, let's talk a bit about its natural history. Where does it come from? So A.mexicanus in its native range is found in this square here in Mexico. Now give me a moment in order to break down this figure as it is a bit complicated. So what looks like gray blobs here are mountain ranges within this area. The blue lines represent river systems, and then the dots here represent the different populations. So you might notice that I have 3 different squares showing different populations of A.mexicanus, as well as the different subpopulations. The reason being is that based on genetic studies, we have found that A.mexicanus appeared in Mesoamerica over 3 distinct invasions. In this red square here, we have what we know as the El Abra populations. These populations appeared in Mesoamerica approximately 8 million years ago, as well as 3.3 million years ago with genetic dating. In these 2 green boxes, we have the Guatemala population and the Micos population. These 2 populations, we believe, arrived 2.1 million years ago based on genetic dating. So they have been here for a very long time, diverging into both surface rivers as well as cave systems. It is hypothesized that the reason that they're found in caves is likely because they fall into these cenotes or sinkholes and are unable to get out. And so this leads to the isolation of the population and eventually these phenotypic structures of troglomorphy that we see. Now let's talk about behavior. This is very close to what I like to do and look at. So A.mexicanus is described as having something known as cavefish behavioral syndrome. This is not a disease, but what it does is describe to us the way that cavefish behave in response to their cave environments. As I described previously, there is typically a lack of schooling. There is increased wall falling behavior compared to surface fish. There is reduced total sleep, difficult to quantify, but related to their circadian rhythms, reduced stress, vibration attraction behavior, or increased behavior when a vibrational stimuli is present in the water, and increased food consumption or hyperphagy. This in combination is likely in response to some of the phenotypic changes that we see. Now let's talk about a concept known as construction and regression. So when you look at this table, we have different features that are highlighted as well as the change. And they say it's either a constructive change or a regressive change. This is not a positive or negative sort of thing, but what it's describing more is if a trait is constructive, it means that it's developing further in order to exploit the environment. Whereas if it's regressive, there is less of that trait present. So for example, if you look at eyes, they say that eyes are regressive in these cave fish, being that they lose a lot of their eye structures. There is actually kind of a primordial eye underneath the skin, but it is no longer functional. And the optic tectum, which is the part of the brain that helps process visual information, is decreased compared to surface fish. Whereas if you look at something like fat content, we would say that it is a constructive change. This means that there is an increase of fat content in the body, likely as a response to store more energy when there is little nutrition in the environment. So we'll be coming back to constructive and regressive evolution. Now, as a sensory ecologist, I love studying sensory systems. So what I gravitate towards in this table are things that relate to how they sense the environment. Things like taste buds, cranial neuromasts, or eyes. So let's talk a little bit about sensory systems. Now, we need to remember as a foundation that sensory systems translate energy into information. You probably know your five senses: sight, smell, touch, taste, and hearing, but I'd be remiss if these were the only five I'd mention. We do have something known as vestibular sense, or knowing balance of your body, and proprioception, which is detecting your body moving in space. Organisms like animals have a lot of these same physiological sensory systems, but some of them are quite different and expanded more than what humans have. For example, sharks have sensors for detecting muscle movement in fish, and snakes have sensors for detecting heat. So they are very diverse in the way that they use sensory systems in order to exploit their environment. For the purpose of A.mexicanus, we will be looking specifically at their visual systems as well as their auditory systems for these studies. So let's break it down again. Quick crash course, what is sound? When I talk about sound, you're probably familiar with this waveform that you see in a physics class. But if we break it down, sound really is the compression and rarefaction of molecules within a medium. So in air or in terrestrial environments, it's the compression and rarefaction of air molecules that transmissions information. Now, I'm sure we're all familiar that the terrestrial environment is a very rich acoustic environment. I'm here talking with you all, and you all are hopefully listening, but underwater environments are equally as diverse. Actually, it is typically suggested that underwater environments, sound in water travels about 4 or 5 times faster underwater than on land. And this is an estimation because there are many factors that affect the way that sound travels. Bioacoustics is a very complex study. Some things that change this include things like salinity, water temperature, other things that are dissolved in the water. But besides all that, you have a very rich array of sound sources. You have organisms that create sound, like whales. You have dolphins. You have things like abiotic factors, like the weather or earthquakes that can create sound. And then you also, of course, highlighted in this figure, anthropogenic sources of a noise, such as boats and pile driving. So safe to say, we agree that the ocean is a very noisy place. However, caves, not so much. So these are actually pictures that are taken from studies where cave fish were collected. And as you can see, they don't have as much going on as maybe an open ocean would. For one, you do not have abiotic factors like the weather that is going to cause noise. You do not have many organisms living in this environment, so there are not going to be a lot of sources of biotic noise. Really, the only sources of noise you typically get in a cave system is the movement of water. So maybe that's a stream or it's water dripping off of the ceiling into the water. So let's talk a little bit about how we detect sound. So I'm sure many of you are familiar with the mammalian ear and how we detect sound. And here I'm going to show you a fish ear, which looks different, but in some ways it's the same. For one, we have these semicircular canals here that look very similar in fish, but when we get down to the inner ear, a very different story is being told. For one, fish have these structures known as otoliths. Otoliths are these calcareous structures, and they serve as a way for fish to detect sound. The way it works is that a fish in water is about the same density as the water. So when sound travels through water, the body of the fish vibrates too. However, because these otoliths are calcareous and are a different substance, they are going to move at a different rate than the rest of the body. So these otoliths sit on top of these hair cells and move at a different rate than the body. And this is what produces the experience of sound in fish. Now, one thing you should note is that fish kind of hear sound differently than we do. For one, they can hear sound pressure, which is the wave or the compression and rarefaction of particles in a medium, but they also sense particle motion. Particle motion basically is the movement of, rather than the wave or the congregation of these particulates, it is the individual movement of the particles that they are able to sense. So I will call this from now on particle motion. All fish can detect particle motion, but not all fish can detect sound pressure, and I'm going to show you why. So if you look at this fish here, you'll notice that we have the inner ear, and then we have the structure known as the Weberian ossicles. The Weberian ossicles are a skeletonized structure that actually connect the inner fish of the ear to the swim bladder. If you don't know what a swim bladder is a gas-filled chamber in the fish that helps with buoyancy in order to change the position in the water. As I told you before, fish typically have the same density as water and move at the same rate, but air doesn't. So this means that this chamber is going to vibrate differently than the body in response to sound. So Ostereophysins, this superorder of fish, have exploited this by creating a connection from the swim bladder to the inner ear. That allows them to detect at a greater rate sound pressure rather than just particle motion. There is a diversity of these types of connections in fish. We have fish that have no air bubble, like sharks and skates. They do not have a swim bladder with which to detect these signals. And then you have Ostereophysins that have this explicit structure connecting the swim bladder to the inner ear. And A.mexicanus so happens to be one of these fish. It does have a Ostereophysic connection. So we know that they likely use sound pressure as well as particle motion when detecting sound. All right, let's talk a little bit about light, since we're going to be talking about visual systems. What is light? Well, light is an electromagnetic spectrum. There are things that we cannot detect as humans, but we are very familiar with visible light. Visible light is known as that spectrum between 400 nanometers to 700 nanometers that allow us to experience vision. Lucky for us, fish have very similar visual structures to us. They have retinas just like we do and lenses, and they actually experience light very similarly. Their range of sight is typically between 400 to 700 nanometers as well. But you need to take into consideration, again, it's a different medium, so they're going to experience this phenomena differently. Light penetrates water in a different manner than it does on land. And you can see here a couple of examples. Here in the open ocean, we see that light can penetrate at a very large depth. It's actually more than 200 meters here, even though this graph is showing 200. Whereas if you look at coastal water or clear fresh water, it penetrates much less. Again, similar to how sound travels differently in water due to different factors, there are many factors that cause light to penetrate water in a different way. And you'll notice here too, one thing I want to point out is while here you get the full spectrum of light going in the open ocean, you would say in this area here in clear fresh water, it's actually more red shifted or shifted towards larger wavelengths of light. We'll be talking about this red shifted phenomena a little later in the talk. And for a little bit of Eye Anatomy 101, I would be remiss not to talk about rods and cones. Basically, cones in your eye help detect color vision. So they help us experience color around the world, whereas rods are more sensitive to light rather than color. It's much less finite than what you would experience with cones, but it still allows us to see a lot more light. And so it's more sensitive to light, we would say. And now for a bit more background on A.mexicanus. I did not tell you the whole story initially because there is actually a recent invasion of A.mexicanus into the US, and this is at the crux of my study. So A.mexicanus we know is native to Mexico, but it was introduced to Texas in the early 1900s. We actually believe that through use as a bait fish, it got released and then was introduced into the Edwards Trinity Aquifer system here, which is quite expansive. We believe, and we have found evidence that there has been subsequent colonization of subterranean habitats as well as surface habitats in this aquifer. Essentially what we were saying, even in not in their native range in a new place, they are colonizing both cave and surface environments, which is incredibly exciting, as it seems to be a prime case study of evolution and divergence all over again. This is the background that I will leave you with. All right, now that we've gone over our background, let's dive into our research question. As I mentioned earlier, we know that A.mexicanus invaded this aquifer about 100 years ago. And our collaborating lab, the McGaw lab, actually did a study in 2020 looking at two populations of fish in this area, specifically in Honey Creek and Honey Creek Cave. Honey Creek Cave is the longest cave known in Texas, and it also has underwater rivers that run through it. Honey Creek is also a surface river. So we found fish, or they found fish in this area and actually did many direct comparisons between the two to see if there was any divergence in phenotype. They did some studies looking at things like length and weight and pigmentation and found significant differences. But what I am most excited about is their neuromass study. So neuromass are a structure that allow for the sensation or to help detect water movement. And you can see that there's two kinds here. You have around the orbit of the eye, these canal neuromass, but I'm interested in the superficial neuromass here. And it's a little hard to see, but you'll see that in the cave fish, there are a lot more neuromass than there are in the surface. It's actually significantly so when tested statistically. This means that there is evidence for potentially rapid phenotypic divergence in less than 100 years in this fish. Now I'm going to take it back quite a long time before this, about 30 years. Okay, so there have been, like I said, previous studies of A.mexicanus about their sensory physiology and many things that it was very exciting when it was discovered. Here you can see actually one of the first studies looking at auditory sensitivity in fish. This was when it was still considered a different species, Astianus jordani and Astianus mexicanus. Let me tell you a little bit about studies of behavior. In the past, we used a lot of conditional training in order to get organisms to tell us / not tell us what they sense. You can't directly ask an animal, but you can train it in such a way that it responds to stimuli in a reliable fashion. Here what we see is a shuttle box with Astianus on one side and another side. And actually what they would do is initially they train the fish by shocking it lightly, and this would cause the fish to learn that it must jump from one to the other over the barrier. Through this training, they were actually able to determine the sensitivity of the fish by playing sound. If it jumped over the barrier, it heard the sound. If it didn't, it likely did not hear that sound. So here's a graph. On the x-axis, we have frequency, and on the y-axis, we have sound pressure. And here's also a little help to show us 3 and 4 are not as important, as these are more relevant to the methodology that was used and looking at background noise, but I want you to pay attention to 1 and 2. 1 is the mean threshold of these cave fish in their hearing sensitivity. 2 is the mean threshold of surface fish. This study found that there were no significant differences between cave and surface fish varieties in their sound pressure sensitivity. However, I would like to note there have actually been no studies directly looking at this sensitivity ever since that 1970 study. All studies currently cite this previous study, and there has been no update or no attempt to look again at the sensory physiology of this fish, which I think leaves a gap in the literature. So what I would like to ask Now that we have these new satellite populations in a new environment, 100 years diverging, is there evidence for divergence of sensory systems between satellite populations? Let's break down this question. So what we know so far about A.mexicanus for their auditory system, we know that cave environments are quieter than surface environments. We also know that from previous studies, losing visual sensitivity may increase the use of other sensory modalities. If you're not dedicating your energy to things like vision, you can use it in other forms, and sound might be one way to better exploit information from your environment. For visual systems, we know that surface fish have eyes, and these troglomorphic cavefish have no eyes. This is because, as shown in previous studies, eyes are energetically costly to maintain. If you're in the dark, you probably don't need them. However, there is no reliable documentation of the transition from eye to eyeless phenotype, because this typically happens over millions of years. This is my hypothesis based on these facts. We hypothesize that cavefish will be more sensitive to sound pressure and particle acceleration than surface fish, because they are using a different sensory modality to better exploit the information of their environment. And we believe that cavefish will be less sensitive in dark adapted light detection than surface fish. You might note here, why am I using dark adapted light detection? This is because when you look at a cave environment, it is likely they will not be exploiting the use of color, as it is typically pretty dark. So for the purpose of this study, we decided to only look at dark adapted fish, or the way they are able to see in dark light environments. So rather than their cones, we're interested in the sensitivity of their rods, which we know, as we previously established, are more sensitive to light levels than the cones. Okay, we have our background done and our research question done. Let's dive into the research that I did. So before we get into methodologies, I do want to talk about our population sampling. As mentioned previously, the other study looked at Honey Creek Cave and Honey Creek Surface, which are found in this red dot location here. It's one geographic region. There is one cave separate from one surface river. So that can be a direct comparison. Then we have actually an additional 3 populations that were found too. We have the San Antonio Zoo population, which actually we see them in a river that flows through the zoo and is part of an exhibit in the zoo. We have San Pedro Springs, where these fish were found, and also Blue Hole. You'll note that these different populations I put in parentheses, things like surface, cave, and cave. These are not necessarily exact descriptions of their environment, as there's still a lot we don't know. For the San Antonio Zoo population, they are in a surface river that goes through the zoo. We can say pretty confidently that's a surface population. However, the Blue Hole cavefish actually are found in these abandoned wells. And depending on the weather, sometimes when it rains, the water table will rise, and these fish, you can see them actually at the surface. At other times of the year, they are very much underground. So can I call that a cave environment or a surface environment? I'm not really sure, but we decided to test them anyways to see if they were leaning in one direction or the other. Okay, let's talk about our first electrophysiology methodology, which is the auditory evoked potential. So electrophysiology in a nutshell is when we take these electrodes and we are measuring a very small amount of voltage in a fish that is represented, or in an animal that is representative of some sort of phenomena that we'd like to see, or stimulus. So in auditory evoked potentials, also known as the auditory brainstem response, this was initially developed to look at hearing disorders in humans with the use of external electrodes on the skin. But we found that you can actually take more direct recordings by placing a needle electrode close to the area of the brain that deals with sound processing. You might not believe me, but this is a minimally invasive procedure, and the fish are totally fine after this. So just rest assured. So what we did is we take one electrode by the brainstem in the auditory processing center, one in a different part of the body. As you know, there is external noise, electrical noise that is produced. So we subtract from this initial recording electrode any noise produced by the body, and then we get a base response for what we believe is detection of sound. What we do is, while in this apparatus, we suspend the fish above a speaker, and then we play different frequencies at different sound pressure levels. We tested each population at frequencies between 0.1 to 4 kilohertz, which is typically what is seen in the literature when looking at sound sensitivity in fish. And then when we test this, we get a result that looks something like this. At the bottom, we have a 5 kilohertz stimulus that was played. And here we have the response from the fish at different decibel levels. And you can appreciate at 119, it is not similar. What we're looking for is a waveform that copies kind of the form that we see in the stimulus. At 119, there's no discernible response, but at 147, we see a pretty strong, robust response here to the stimulus. This is shown, again, as you can appreciate this visually, and also we use a statistical method in order to determine whether it is a response or not a response. Now I'm going to show you some of the data that we collected. So you are aware throughout this talk, we're going to be using the same colors, so that way we don't get confused. So here we have our first table. Here on the x-axis, we have frequency in kilohertz that was tested, and on the y-axis, we have sound pressure. In green, we have Honey Creek Cave, and in red, we have Honey Creek Surface. The way we generated this graph is that for all the groups, we averaged the sound pressure level that was needed to elicit a response at different frequencies. We then plotted this threshold, average threshold, across each frequency, and we actually did individual t-tests at every single frequency level tested. So every data point represents a t-test that was conducted in order to determine the difference between the averages of the two groups. In this case, because the data was not normal, we decided to do a Welch's t-test. The asterisks indicate significant results, and as you can see here, the overall trend shows that Honey Creek Cave had more, and the way you read this as well, is that the farther down on the graph it is, the more sensitive the fish is, because it's a quieter sound that is needed to elicit a response. So they are more sensitive. So here in green, you see that Honey Creek Cave is more sensitive than Honey Creek Surface to most of the frequencies tested. And this is significantly so in this range here, which is about 0.5, 6, 7, and 8 kilohertz. This is promising because this correlates with a lot of other data we see in fish, as fish are typically more sensitive at lower frequency levels than higher frequency levels. So that is our result for our first two populations tested. Now let's look at our other three populations. As you can see here, we have three populations rather than two, so we cannot run a t-test. Instead, we ran a Kruskal-Wallis, ANOVA, and then we did post hoc testing in order to see significant differences between individual groups. And what we found here is that actually we did have some significant results, mostly between the San Antonio Zoo surface fish and the Blue Hole Cave fish, in a similar frequency range to that of the Honey Creek Cave and surface fish. Our conclusions from this portion of the study is that Honey Creek Cave fish are more sensitive to sound pressure than Honey Creek surface, and that Blue Hole Cave fish are more sensitive to sound pressure than San Antonio Zoo fish. Let's move on to our next methodology, particle acceleration. Now, in particle acceleration, we're not determining sound pressure, but actually the movement of particles. So we use this cool tool called an accelerometer. This accelerometer measures movement in the X, Y, and Z directions. So what we do is we play the same sound that we would at the fish when testing sound pressure and see what kind of reading the accelerometer gives us. And here are the results from that study. Now here you can see there's an appreciably bigger difference between the cave and surface fish than there was in the sound pressure at many, many more frequency levels. So here we would say Honey Creek Cave is significantly different and much more sensitive to particle acceleration than Honey Creek surface. However, for these three other populations of San Antonio Zoo, San Pedro Springs, and Blue Hole, we found no significant differences in particle acceleration. So our conclusions from this portion of the study are that Honey Creek Cave fish are more sensitive to particle acceleration than Honey Creek surface, and that there were no significant differences in particle acceleration between San Antonio Zoo, San Pedro Springs, and Blue Hole. Now we'll move on to our last methodology of this study, which is electroretinography. Now, this is a bit of a complicated graph, and I'm going to zero in on what's important, but you should know that basically electroretinography is similar to the auditory but evoked potential technique in that we're putting electrodes in a place where a sensory integration is taking place. So we actually put electrodes in the eye, a needle electrode in the eye and in another portion of the body. This is minimally invasive and doesn't harm the fish. And what we do as we zero in here is we take a monochromatic light source between 400 and 700 nanometers, as we're interested across the range where it is more or less sensitive. We put it through this optic light pipe and flash it into the eye of the fish. And then when we send the stimulus through the machine. We then pick up whatever response the fish gives us. It goes through a signal amplifier, and we get a response that looks something like this. This is a good example of electroretinography, an actual response from a fish that we used in this study. How you read it here is on the x-axis, you have amplitude, millivolts, and the y-axis, you have time. This black bar here represents the stimulus that was played to the fish. So this is a 200 millisecond stimulus of a light source, and this is the response that we get. One thing I will note for electroretinography is you have two components of the wave. You have something called an A wave and a B wave. An A wave is the response of the cones to the light, which we do not see here because it is dark adapted, exactly what we wanted. Instead, we see this B wave, which shows us the rods in action, detecting light. And here are our results from this portion of the study. So on first appearance, you would say, wow, that is a significant difference between the two populations. However, we found it was only significantly different at 530 nanometers. Here, and just to orient you to this graph, on the x-axis, we have the wavelength tested in nanometers, and on the y-axis, we have irradiance. So here we would say there was a significant difference between Honey Creek Cave and Honey Creek surface, but only at 530 nanometers. In this case, the way you read this graph is the farther down it is, the less sensitive it is. And the farther up it is, the more sensitive it is. So Honey Creek Cave is actually less sensitive than Honey Creek surface. As we saw with particle acceleration, when looking at the graph here, there were no significant differences between the three additional populations of San Antonio Zoo, San Pedro Springs Cave, and Blue Hole Cave. So our conclusions for electroretinography are that Honey Creek Cave are less sensitive to light in dark adapted conditions than Honey Creek surface at 530 nanometers, while there were no significant differences in sensitivity to light in dark adapted fish between San Antonio Zoo, San Pedro Springs, and Blue Hole. Now that we've gone over all of the methodology used, why don't we wrap this all up and see what we see. So in summary, just to reiterate the results, Honey Creek cave fish are more sensitive to sound and particle acceleration than Honey Creek surface fish, and Honey Creek cave fish have less dark adapted visual sensitivity than Honey Creek fish or surface fish. And while we did see some differences between San Antonio Zoo, San Pedro Springs, and Blue Hole, they were not nearly as robust or dramatic as the difference between Honey Creek Cave and surface. Now, there's a word I've been avoiding throughout this talk. It's the E word, the E word being evolution. So you might be wondering, well, Maya, how can you tell, is it evolution or is it plasticity? This is a very good question, so let's break it down again. Evolution is defined as genetic changes that are heritable across generations. Plasticity, on the other hand, are not genetic changes and are not heritable. And one good example I'm showing here is that of carotenoid coloration. So you might be surprised to find that flamingos are not born pink, they're actually gray. And through their diet, eating things like shrimp and algae, they get beta carotenes that create this pink coloration. If I put these two birds together in a room, you might say those are two different species, but it's not. It literally has to do with behavior and with phenotypic plasticity. So is this what's happening in our fish? Let's go to our future directions and I'll break that down a little more for you. So for future directions, one thing we would like to do is to better quantify our cave environment. Cave environments are very cryptic, typically hard to access and reach, and there's just not a lot we know about them. So one thing that we can easily do, now that we know that this cave is accessible, is we can deploy hydrophones, which detect sound underwater, as well as light loggers that detect the intensity of light in these environments. This will likely help us better explain what we are seeing in our electroretinography assays, as well as our sound pressure assays. Another thing that we're going to do, very excited about this, the Magal Lab is very gracious in giving us these awesome troglomorphic fish, the native populations in Mexico. We have molino fish and pachon fish that we will be testing. Obviously not electroretinography as they don't have eyes, but we can test sound pressure and particle acceleration. In addition to this troglomorphic variation, we're also going to get some surface fish, and we can test those the electroretinography assay for visual sensitivity, as well as sound pressure and particle acceleration. Okay, for the plasticity portion, we're lucky enough that the lab in the Twin Cities was able to spawn and rear some of the wild-caught satellite populations for us. They've been growing them for about 1.5 years, and coming up in the spring, I will be doing additional assays in order to test these young fish and see if there are differences in their sound pressure detection, particle acceleration, and visual sensitivity. So we can answer once and for all, is it evolution or is it phenotypic plasticity? And last but not least, for you statistic nerds out there, one thing you might note is, Maya, when you're testing all these different levels, you're going to be losing statistical power. And I agree. It's a little bit annoying to have to do a test at every single level tested. So one thing that I'm actually working with Dr. Ted Ozerski on is comparing sensitivity curves using generalized additive models. What we do here is, rather than pick apart every single level that we tested, we actually look at the curve. Here we have the curves showing using additive models. And then we have our 95% confidence intervals. And then we have this area in white here. The difference between these two curves will tell us that there is a significant difference between these two populations in a different way than we would just doing the t-tests and ANOVAs on the individual levels. And that is my future directions from here on out. So this is the conclusion of my talk. Thank you so much for attending. Before we get going, I do have many acknowledgements to give out, so please bear with me. For one, I would like to thank my wonderful committee members. For one, Dr. Alan Menzinger for being a wonderful advisor, as well as Dr. Thomas Harabik and Dr. Amanda Klein for agreeing to be on my committee. This work would not have been possible without other members of the Menzinger Lab, including, for one, Nolan Michaels, who has been a wonderful mentor, and who's currently not here, but worked for a very long time, Roz Putlin, who taught me the auditory evoked potential technique. And of course, I couldn't ignore the wonderful undergrads in the corner here, Amanda Kistner and Ty Tate, who have been very helpful in collecting data. Amanda is actually going to be running a behavioral study of sound, and Ty helped me collect electroretinography data. And all three of us will be going to a national meeting in the spring, the Society for Integrative and Comparative Biology, and I'm very thrilled. In addition, of course, to our lab, we have the McGaw lab. Without their help, this would not have been possible. My thesis wouldn't have happened. So I have to thank, of course, Dr. Suzanne McGaw, Dathan Swanson, which is her master's student, who helped transport the fish and teach me about animal husbandry, and Dr. Andy Glesenkamp. He is actually the director of conservation at the San Antonio Zoo, and he collected our satellite population fish and got us those organisms that we needed. Now, let's talk a little bit about the Integrated Biosciences program, which is the whole reason why I'm here. Yeah, so for one, I would like to thank Stormy, because without her support, she is the first person who met me when I landed in the ice cold wasteland of Duluth in winter and assured me that I would be okay, even though I was from California. Dr. Jennifer Liang was my first contact for this program. And honestly, I think I share a lot of her vision in the things that she does here. And she is a large part of the reason why I decided to stick around. And of course, the wonderful IBS cohort, which are sitting in the 2 rows in front of me. You guys are fantastic and are a wonderful support system. I would like to financially thank the Dove Fellowship, who supported me for 1 year. And that really allowed me to get ready to do research and transition from working life to research life, which is quite an ordeal when you've been out of school for 2 years. And of course, friends, the Enriquez family, and I'd say family in general. As shown here by this wonderful illustration by my brother, it's truly family that gets us through this. So thank you for my genetic family as well as my chosen family. And thank you for listening to this talk.
We'll start with questions from the undergraduates.
Yeah.
Well, I was wondering, so you're talking with the cavefish, they need less sleep. Is that due to like reduced biological stress? Like they don't have eyes that put energy to that and there's not much competition?
Yeah, so that's actually awesome question, because that's what I'm going to be doing for my PhD. We're going to be looking at circadian rhythms and sleep, which is hard to quantify in fish because they have no eyelids. But we believe the reason that they sleep more, for one, is that their circadian rhythm is out of whack. There are no night or day cycles in cave systems. And actually, it's very interesting. Your circadian rhythm is distributed throughout your body, not only for sleep, but also for things like DNA repair and things like that. So it almost seems in some cases that they're living in continuous daylight. Their circadian rhythm is like off kilter in such a way that there's no shifting, but just one solid, like awake all the time almost. It's very difficult to quantify, and that is going to be what I will dedicate the next 5-7 years of my life to. So I'll get back to you on that. But great question. Yes.
I guess one last a little bit, since they need to eat less than diet, stuff like that, do you think potential study of like exclusion would be like that thing would be, or like different?
Yes, absolutely. Actually, we were kind of hoping to workshop a study looking at how they use energy in our fantastic swim tunnel that we have on campus. We purchased a swim tunnel with student tech fees. Thank you, Animal Physiology. And we are interested in looking at how they use energy by seeing oxygen consumption in this environment. As far as do we look at that in general? Yes, we definitely do. Cavefish are weird in that they are both can go a long time for not eating, and when they eat, they actually have hyperphagy where they eat a lot. They're actually a very good model for use in diabetic studies because of the way they have this metabolic syndrome that is beneficial in their cave environment. So good thought. I'm sure someone's done it, but yes, much more to explore.
Roger, students.
Hi. Yes, you.
Two more questions. First, curiosity, has there been any evidence of synesthesia in fish? And then what measures do you use to control for whatever you, or the organisms, individuals that you're studying having normal senses?
Both very good question. One, I do not know if fish experience synesthesia. The hard thing about doing fish sensory work is you can't just ask the fish, hey, what's up? I'm sure there would be a way to measure that indirectly, but I don't know what that is. The second question being, how do you control for individual differences is a very good question. I don't know quite how I would answer that. I think that's the crux of a lot of organismal research is that you don't know what you're going to get. So you increase your sample size in order to account for differences and average, but you're absolutely right. One thing that I will say, a thing that's kind of a bugbear that's been bothering me about this work is all our cave troglomorphic fish that we're going to get are all lab reared fish. And I do wonder, I'm wondering like, how do like lab environments affect their hearing and things like that? I would love to get my hands on native fish from their native environment.
Maybe going off that, is there any plans, maybe collaboratively or with you, to do genetic analysis of cave versus surface populations? Because that could also establish like whether it's true evolution or just plasticity between.
Right. So I'm very excited about this PhD because I'll be doing a completely new tool set of things here. I've done a lot of sensory phys and behavior, and that's actually all genetic work is what I'm going to be doing. So I'm sure work like that has been done before. The genetic work I'll be doing is actually crossing individuals F1s and F2s between cave and surface. And then we're going to be looking at changes in behavior, i.e. their circadian rhythms, and looking at that. So I know that there's a lot of work in that field and there's a lot I'm going to learn. So, but very good question. Again, okay.
It's totally different though.
Okay.
Why didn't you compare the Honey Creek and cave and surface populations with the Blue Hole and Sandpaper River, the other populations of that?
That is a very good question. So we did establish ahead of time what we wanted to compare. I think there's a bit of a danger in comparing everything and seeing what happens, kind of like throwing darts on a wall. So we thought it would be most parsimonious if we compare the 2 geographic regions as they've been compared before. The 3 other ones, those are honestly shots in the dark. That's why we grouped them together. But good question. Hi, Nolan.
Hello. Well, first off, great talk. And also, I got kind of interested. You mentioned this redshifted idea. When I looked at your secondary graph, it kind of showed that I think the sandpaper population would have a more broad spectrum. I wonder if you just have any ideas about that.
You're talking about this wave, this curvature here, right?
Yes.
Yes. So again, I guess I'm going to say to my defense, we don't know a lot about the environments of these fish. I know for a fact that the Blue Hole Cave has a very transitional environment or ephemeral environment where it's like sometimes surface, sometimes cave. And we haven't really quantified these environments very well for San Pedro Springs. So I would say this is likely in response to something in the environment. And that's why we're hoping to deploy these light loggers and hydrophones in order to better quantify the environments. So we could answer a few more questions as to why. So great question. Thank you.
Questions from anyone?
Yeah.
When you mentioned that the two different morphs can interbreed, is that just in the lab or has that been shown to occur naturally?
Awesome question. Yes, so we have done that in the lab for a lot of research reasons, but there are some populations where you do have regular gene flow. Sometimes during flooding events or other weather events, you actually get fish pushed into the cave. And so there are certain populations where absolutely not. You only see troglomorphic fish and river fish, surface fish, but a couple of them do have hybrid varieties there. They're not nearly as many as the troglomorphic, true troglomorphic varieties, but yes, it does happen naturally. Whoever would like to go first.
What happens to the cranial organ and melatonin?
Yes, okay, let's talk about that. So as we know, let's get a good picture of a cavefish, but the cavefish have no eyes. However, the cavefish do have something. Let me get to a cavefish picture. Here, we're just gonna use this. So cavefish, no eyes. But one thing we do know is that studies have shown that they can use their pineal gland to detect light. It does detect light. It does function as a source of light detection, which is located around here. As far as regulation, it goes into a bit of the question of like circadian rhythms. So in these fish, you can almost describe it as being bathed in consistent daylight. It's almost like it's constantly exposed to light, even though there is no light in their environment. And we believe this might be an adaptation or a shift in their circadian rhythm in order to still do things like DNA repair and other things that are required for survival. We actually did talk briefly about potentially trying to record sensation, like do electrophysiology on the pineal gland, but that, I don't know, we'll see. That might be kind of cool. Something that hasn't been done before. Yeah, the one thing that is nice about the methodologies used is our fish, unless something did not go well, all our fish survived the methodology. I really do mean it's not invasive. They're totally fine afterwards, and they're under anesthesia when they do it. So, and I believe you had a question as well.
Yeah, I'm just curious about the hybrids. It seems like you can gain a lot of information about your question of environment versus genetics by the phenotype of your hybrids.
Correct.
And so what are the phenotypes of the hybrids?
Of course, here, I don't have a picture of that, but I'll show you a picture of them together and we can talk about what it looks like. So typically in the cave varieties, they're typically much larger actually, and they also have more fatty deposits, which is why they look kind of yellow versus these guys. In the hybridization, typically you can see they have lighter coloration, but still some coloration. So they have some pigmentation. And they actually have an eye, but there's actually some skin covering it. So you only really see the pupil. So they do have eyes and they do have pigmentation. It's just a little bit more cave-like rather than surface-like. So that's why you can do tests on vision in these hybrid varieties. There is a retina, a functional retina and everything like that. But yes, I will make sure to have a picture of that for next time.
Yeah, and it just seems like the inference, if they have intermediate phenotype, when you hybridize them, is that it is genetic.
Right. And I believe through this hybridization, like you get a mix, you get some of them that look like surface, some of them that look like cave, and some of them look like in between. And that's going to be a lot of, actually, I think I have like 100 fish that we have to rear where I'm going next. So yeah, there's going to be looking at a lot of F1s and F2s in order to narrow down questions. Sure.
So the Texas fish, you mentioned this troglodytic morph might have evolved within fewer than 100 years.
Right.
Has genetic work been done to show that that was a single introduction? And there
Yes.
was a separate introduction to the surface and cave morphs?
Okay, I see what you mean. Here, let me just go to this slide. So here's showing again a little bit of difference between cave and surface. We do not know a lot about these surface populations. This is actually the first publicized study on satellite populations at all. So I'm not sure. I think that would be a good future direction zone. It'd be something that should be quantified because we know approximately when fish arrived, but more we know it for this population in particular. The other ones still unknown.
All right, it's 4 o'clock. Thank you all for coming.